So it looks like we should get started now. So first of all, thanks for all joining us again at our third webinar, our FEDM's Data and Model Management webinar series. It's good to see quite a few people back as well. I'm Natalie Stanford and I'm the FEDM Community Lead, and I'll be your host today. Now, FEDM is an initiative to support researchers, students, trainers, funders, and publishers. In fact, anyone interested in data and model management in better data management practices. We offer advice, expertise, and training which make managing your data and models in life sciences easier. And you can find more about us on our website at www.fair-dom.org, or you can follow us on Twitter at fairdom_EU. And if you have any questions regarding FEDM, you can get in touch with me at community@fair-dom.org. Now, before we go into the webinar today, we've just got a quick announcement. So FEDM's going to be hosting its first user meeting as a satellite meeting of the International Conference on Systems Biology. And this meeting will be a full-day meeting in Barcelona on the 15th of September. The meeting will be for anyone using or interested in using FEDM software or services. And during the meeting, you'll hear from high-profile research labs about how they structure their laboratory pipeline using FEDM software, and also how they integrate other software such as eLab notebooks. There'll also be portions of the day set aside for breakouts and discussions based on topics of interest from attendees. The expected topics, we think, will cover things like integrating FEDM software with other software, using FEDM, and publishing citable data with FEDM. If you want more information, you can visit our website and you'll find a flyer on the front page which will take you to a news item. Now, back to the webinar, and today we're here with our guest, Philippe Rocca-Serra, and he's from the Oxford E-Research Centre in the United Kingdom. Good afternoon, Philippe.
Hello, good afternoon, Natalie. Good afternoon to everyone.
Yeah, good to have you here.
Thank you.
Philippe has 15 years of experience supporting data generators and service providers to generate reproducible research. And he started this work at the European Bioinformatics Institute in Hingston. And he's now a senior research lecturer in Oxford's E-Research Centre and a consultant for Hoffmann La Roche Healthcare Company. He's currently co-leading several European and US initiatives covering knowledge management in clinical settings, and he also contributes to the Elixir UK node as part of 2 NIH Big Data to Knowledge initiatives. Today we'll be talking through examples of ISA and biosharing resources. If you have any questions during his talk, you can use in your control panel, you can ask questions using the questions tab. And we can come to these at the end of the talk. Or if you want, you can raise your hand and then we can unmute your mic so you can ask questions later. So I guess we'll hand over to you then, Philippe.
Okay, well, thanks very much for the nice introduction. And thank you very much for giving us the opportunity to present to the FEDM group. Really, that's great to be here this afternoon. So first of all, I hope you can see my screen. Let me know if not.
It's looking good so far.
Yeah?
Yeah, perfect.
Okay, excellent. So I will be dividing my talk into 3 parts, essentially. One will be just to give you a bit of a view of the Biosharing catalog. Then I will tell you about the history and about the ISA project, where it started from, and why and where it's going, essentially. And then I will touch upon recent developments that we've been involved in surrounding the application of such technologies or some principles to creating a e-infrastructure. This is a project recently funded. So I'll start with this kind of observation that basically we've got pressure from everywhere about data sharing. There is almost not a single week where there is no editorial in a high-profile paper asking for this kind of a release of data or algorithm. This is last year in Nature, another one a month later, nearly again asking funders this time to put some pressure on wet lab biologists or anyone doing data science and producing data to grant access to this information. And when I'm saying pressure from all sides, obviously there are calls like this, but the funding agencies are starting to put this kind of pressure as well. So this is the NIH policy, and as you probably know, there is kind of incentive to release your data under 6 months for all the NIH program funded research. The regulatory agencies as well, such as the FDA in the US, are asking now for all submissions to be made in CDISC format for the clinical trial data. So again, that's a good kind of big pressure. Journals obviously have their own data availability policy. Here I'm showing +1, just because I've been interacting with the journal as I was working with the metabolite repository, trying to get my hands on some data itself, and it's kind of a nice interaction that we had. And also I can cite Giga Science from BMC, which is a publication geared toward large data sets and genomic science that also has for data availability. And of course, Scientific Data, which is a recent data journal released by the Nature Publishing Group. But it's not only those key players, so funders, regulatory agencies, and publishers, but also the public is asking for this kind of data. And there is a growing collection of, in a way, patient advocacy groups are asking for clinical trial data to be at least registered, so people can at least be aware of the various trials that are taking place. And this is, in a way, an illustration about how access to data is, and awareness of data is changing our way to relate to scientific work. So, of course, all these people speak softly, and some of them carry a very big stick, and we have to be careful as scientists that we are ready for that extra work. And I think that's talking of experience. When I was doing wet lab work many years ago doing sequencing, the first time I had to submit data to NCBI, it was a bit of a shock. So it's a bit of a shock because I realized it was only for a few sequences, still a significant amount of work. Of course, today the tools have evolved, you have pipelines to do this, but as new data types are coming along, the similar process has to be experienced, and people go through the pains of testing tools and refining them during submission. So, and when you're a wet lab scientist, sometimes it's kind of difficult to know where you are. So, the one thing that we have to bear in mind is that besides this kind of pressure from publisher to submit the data, there is another, not danger, but things that we need to be aware of. The fact that the main reason for disclosing the data is that it can be evaluated or reused, and certainly you don't want to end up on the retraction watch as part of the data reuse. So one aspect to bear in mind is that when you do data management, it's also to ensure that the quality of the data that you're collecting is really captured. There is no big problem that you have overlooked, and you're careful to keep points. And the fact that such blog exists, and it has raised attention because it's also overspilling in the New York Times. So it's interesting to see that even big media are taking this very, very seriously and can make the headline that scientific output is being watched and reviewed and assessed, and therefore, in a way, everything that is related to data management will become critical, and we need to have a better way to help the scientists in their daily exercise of science to deliver on this front. So, of course, the one consistent message is that we need to remove the obstacles to data access, but for a lot of people, this is the feeling I had, at least when I started submitting data. It's like, where should I start? What are the tools to help me? And there are so many formats, so many standards, and so many data types, of course, and we need to make sure that we get help, and the experience can be very, very depressing at times. So, and this is really why we decided to create this biosharing catalog. To begin with, life science, there are about more than 600 content standards, and we can structure them in different types. There is a kind of typology of these resources, whether we are looking at syntax, terminologies, or simply guidelines. So some of you will be familiar with some of the keywords that are showing on this slide. But really, what we want us to remember is that all these standards are usually developed by a community of people who are really keen to make sure that key information is reported. And we can distinguish also the kind of organization that are behind the standards. There are very well organized bodies, which are really standard developing organization, almost industry strength. So ISO is well known, CDISC, W3C, or HR7. These are the standard organization, really well-established bodies. And then there are the grassroots on the right-hand side, which maybe are closer to the wet lab or to the kind of research academic areas. And there you will have efforts such as the Genome Standard Consortium or the Proteomics Standard Initiative, the Metabolomics Society, Combine, and which also operate in that space. And then we need to reconcile these two. This also explains the dynamics of the world of standards, where basically there is a certain level of fragmentation, and we still need to build a landscape, a map to survey all these landscape. So that's really the main raison d'etre of biosharing, just to connect all these resources to provide this map of standards in life science, so that it can be harnessed as a tool to help people to building their data management plans, for instance, or being aware, being aware of the resources that are already out there to support their work. And I will dig deeper, but you can see already the kind of overlap between the standards and then the relation that can be made between databases that are actually implementing some of the standards or policies that are advocated by funding agencies, journals, and societies. So, in a way, this is the kind of use case and scenario that we want to support with biosharing. So, if I'm a research scientist, I'd like to know, first of all, if my funding agency, I'm on this bottom area here, if my funding agency recommends any specific standard. And especially if I'm working in a particular field, such as clinical trial data or toxicological exposure, what I should be aware of. And then again, if I'm doing metagenomics, do I need to be aware of any database that is the recipient of such data set and which guidelines they would be implementing? Conversely, for funding agencies and journals, they also need help somehow. I mean, it's difficult for research scientists, but it's also difficult for those players in the life cycle of data science, for them to know exactly which are the key resources. So that's again a role for biosharing to assist, as it is for curators and developers when they need to go back to a place where they could say, I'd like to obtain information about the latest version of the standards or the terminology, for instance, related to cell-based assays. So really, we try to support these different people, this different group of users, and really this is why biosharing exists. So you have a basic search interface, which organize, as I mentioned, basically the standards or the resources for data management according to three main axes. So terminologies, syntax, and reporting guidelines. And each of the standards has been tagged with a number of internal ontology that we maintain, so that we can index and organize this information in a more effective way. What we have recently added to the biosharing interface is the kind of recommender system, whereby you come to the interface and using the, I hope my screenshots are coming through correctly, but you can basically with the advanced search start selecting the kind of standard that you would like, or policy or database that you want to search for, and then restrict the elements by the kind of data type you're most interested in. And the tool will therefore provide you with a list of resources that are most specific to meet your requirement. And for each of the standards, we also track the evolution of the resources. So some of the records you will see, consulting biosharing, are aging and can be deprecated, and they will be shown like with this strike-through type face. But also, if that occurs, we also indicate whether it has been superseded by a format. In that example, we have MZ data format that has been superseded by MZ mark ML format, say, and this is clearly indicated from the records. So one of the other tasks that we want to attach to biosharing is to build a kind of metrics of success of these different efforts. And one metrics of success is to check the uptake of a standard in databases which are actually storing data of the same type. Here, I'm just showing the kind of relation that we captured between the Miami reporting guideline on the right, with the Miami notation markup language, minimal, that is a format, an XML format supported by the gene expression observers at the NCBI. And therefore, you can really see the kind of interdependency between these elements. And I'm just giving this example for Geo, but we have the same kind of layout that relates Miami to MageTab and ArrayExpress at the API. And this is, in a way, a good metrics, and we sort the various databases that are stored in the biosharing catalog, which has these two components, the component of standards and the component of databases, so that we can have a feel for which are the most prevalent file formats and nomenclature terminologies which are used in the biodomains. So this is a bit a busy slide, I realize that, but it's just to show you the kind of different kind of information that you can find, and to insist again on the relationship between the standard records and the database record, which make this kind of two big silos behind the biosharing front end. So we have the isotab format, for instance, which is implemented in the metabolite repository at EMBI EBI. We have information about the person who maintains this record, which is myself in that instance, and various kind of other relation connectivity graph that details the information about the relation to other formats, and also identify which resources are currently using the format. So another slide, really this is just a summary of the key information that how to use it when you consult the biosharing catalog. And I hope that you will find in these personas one that really matches your need. So the catalog is currently being evaluated to become an Elixir resource. I think that would be a very nice addition to the collection of resources that are being operated, and could be used as a kind of guiding principle and control tower for also identifying gaps in coverage where the community needs to make effort in terms of providing better tools, better formats, or better terminologies to support the work of scientists. So now I'll flip to another, to the second part of the presentation, which is essentially telling you a bit more about the work that we've been doing for the last 10 years about the investigation study assay model. And again, this is an effort trying to capture as much information as possible when confronted with studies which use an array of assay types to determine the molecular phenotype or the phenotypes of biological systems. And in a way also it's to move the data collection from the kind of paper and pen based approach to the full Monty, I would say, data management in the 21st century would be linked data and in the cloud, basically. But in the middle, there is a kind of transition phase that we need to go through, which allows to provide a semi-structured information, and this was by the means of spreadsheet and tables. And so the approach is important because it was a way to maximize a fairly widely used practice of using spreadsheet in the wild, and at the same time try to retrofit or bring awareness about the need to annotate and to limit free text in those reports that were generated by scientists. So how do we help this task and make it almost invisible? So that's the kind of a project. So really what started the project is that we were confronted at a very early stage when we were working back at TNDLBI in the Macquarie Express group. We were confronted with people doing toxicogenomics, and they were using microarrays, but they were also using already metabolomics and proteomics and range of classic biochemistry analysis and histopathology as well. So the reports were very broad, very thick in a way, and also the key thing is that it didn't match with the topology of the repositories the way they were established, which were very much assay-centric. And what we realized is that we were losing, when depositing the data in repositories, we were losing the connection to which sample belonged to, derived from which patient or which animal. And that was, I think, a kind of missed opportunity. And so that's really what drove the creation of the format, which in a way borrowed a lot from existing resources. We simply borrowed and reused what already existed to make sure that the acceptance would be fairly straightforward, so the uptake would be easier, and we could reuse existing tools as well, and show compatibility. That was the main purpose, so that this compatibility could in turn ensure that we could convert to the format the repositories accepted, and this was providing a service to the community as they, in a sense, we would provide a unified interface, but we would take care of the burden of converting to the specific formats required by the repositories. So the format turned out to be a success, and the format was picked up by the MBL metabolite repositories, but also by the Harvard Stem Cell Discovery Engine and Institute. It was also chosen by 2 publishing houses and to Scientific Data and Giga Science, and also used now by the COPO project and also the Phenomenal project. I will tell you a bit more in a few minutes. So this ecosystem of tools started with on the left-hand side with Isaac Configurator, which allows you to provide a number of restrictions and requirements in the tables that we produce, which are then used to create a user interface in Isaac Creator, which is a Java standalone tool, and which produces ISATAB documents, which can then be converted to a number of formats, so MageTab to REXPRESS, SRA, XML for ENA, SRA deposition, will also provide XML for deposition to proteomic repository at EBI. The interesting bit is that going back to the key feature of Isaac Creator is that it was a spreadsheet look and feel, and then what we had a request from users, like how it would be great if we had collaborative editing features. So then we developed Automaton, I will mention in a moment. So this is the key feature that made a success of Isaac Creator. First, it was a spreadsheet-like interface. So that was, in a way, listening to the needs or the feedback of the users. We like our spreadsheet, it's a natural format, why change it? So we just follow the advice. The one thing that we added is the ability to have ontology lookup search and tagging, automatic tagging, QR code generation, so that we had the possibility of creating tags that could be used by systems, hardware, also visualization components, which produces a glyph-based representation of the experiments, which can be used for publication figures, because they provide a kind of key information about the experimental design, but summarized in a small icon glyph, which is meaningful and whose caption can be automated. So these were the key features of the ISAC reader. And it was so successful, as I said, that this tool, we had feedback, well, it's nice, but I don't like the standalone tool with like a web-based interface. So here we had to optimize, and we thought, okay, what would be really cool if we could connect Google Spreadsheet, which provides a collaborative feature out of the box. We don't have to think or engineer it, it's there already, with the ontology lookup service capabilities that we had in Automaton. And this is just what we did. We wired up to system, and basically, thanks to the configurations that we're able to export in Excel from ISAC configurator tool, we were also able to load those documents directly in Google Spreadsheet, activate the Google app Automaton, and I have this ability to right away have people with collaborative editing capabilities and semantic tagging. So, again, it's a FEDOM pro seminar, so we have to think about these terms. And this is where also we started to reach out to publishers. And we were approached by Science Nature Publishing Group, and they selected a format. And then we worked, again, about data dissemination, trying to find incentive to make people deposit their data, or at least disclose some of the information. And one way to do this, again, has been how do we make sure that once people have gone the extra mile to take care of, show some love to their metadata and provide this, then we should reward them by providing a nice visualization functionality that somehow can help the impact of their work. And this is the scientific data ISA Explorer, which is a very thin layer UI on a file system, but it just provides a kind of insight into how this development can go a long way in enticing people to do a bit more. So here, this is just a kind of layout of the different data sets that have been presented. But if we drill into one particular element, we use the metadata to have a kind of insight into the key variation or source of variation that are available in that particular data set. This one is an RNA-seq transcription profile looking at different organs and different age and different sex in fish or rats, basically. And so the organism has no variation, it's all rat, but here already we got a feel with this ring representation of other kind of changes that occur. So the next elements that we listening to the people working with us is that okay, it's all very nice, the UI, it's all very nice, this kind of feature, but we would like to interact with modern tools through the API. So that was more a request from developers. How do we manipulate ISA format? And this is really where we started thinking about developing a programmatic interface to allow people to edit and deal with the small data that has to come always with the big data. And so this is a page of the GitHub repo that we maintain. I just wanted, it sounds anodyne, but I just wanted to point to this set of tags which show that the kind of build process that we, organization of the team, that every push that is, or commit that is made, run through the build process, and we check that everything runs smoothly. So the unit testing, and in terms of software development, just to give you this insight, we need to provide robust software, and also it just builds the ability to have confidence in what is released. You've got information about the licenses, and you can contribute code or push for misuse directly there. So not only have we now developed this API, we've also used GitHub to keep track of the ISA configurations, which are a very powerful mean to just document any changes to the configurations and provide this kind of repository out of the box. We don't have to maintain so much infrastructure, yet we provide the ability to really track the evolution of this configuration while providing the API a solid point to retrieve those documents before running, for instance, a validation. And of course, we are building this now to have microservices that can be put in the cloud. So this is a kind of a recent work. If you want to read more about this work, you can go to this to our blog. And so cloudy with a chance of meatballs, MTBLS corresponds to the keyword for the accession number in metabolites. And this is where I'm bringing now the work on phenomenal. So the Docker containers for the ISA converters and both from JSON to tab and back, and also capability to run the API in Jupyter, which is a kind of IPattern notebook, is kind of interesting because it demonstrates first the functionality of the API, but also the ability to deploy those things as microservices, and that can be really built in a bigger framework. And why are we doing this? We're doing this because we are, as part of the Phenomenal project, an H2020 project using, grouping all this premier group working in the field of metabolomics to build a new infrastructure supporting the science behind metabolomics. So really the key objectives here are to deliver and operate an infrastructure. We need to reuse open source tools as much as we can, and we need to provide infrastructure that can scale up to be able to work with big data. But our particular work for ISA team is to deliver all the tools that allow the manipulation of experimental metadata, and that should always accompany these big files. So that's really the last few slides. I will be presenting this kind of infrastructure where we are, in a way, providing these Dockerized versions of different tools, including Galaxy, because this is what will be powering a number of workflows. But in a way, this is the kind of also information that from the source code repositories, we start building the tools. All the testing occurs within Jenkins, so we've got a constant report about what breaks and doesn't break, and then we can register the different microservices either into Docker Hub, the official one, or private phenomenal specific elements. And the orchestration is done through a different tool. So I think I'm overshooting already in terms of time, so I'll skip this slide. But basically the key point is that we want to be able to deploy those services in different contexts, whether it's a big center or a smaller center, but using this kind of private virtual infrastructure that will be reusing a number of components from public efforts and open source software. Going back to the FAIR information, this is where of particular interest to us is the publication workflow. Once people have done the analysis and we have a workflow which are aware of the key information that should be recorded, then we can make sure that just by operating an analysis workflow, we have enough information stored in ISA documents, annotated with proper ontologies. And this is why I mentioned Stato here, because we feel that there is a bit of a gap in terms of how statistical results should be reported. So we did a bit more efforts to develop semantic artifacts that could also support a good reporting of statistical results. I'm just showing two screenshots about Stato. You can see more by going over on the specific dedicated website, statoontology.org. But these are the kind of use cases that justify the development of the resource. And the ontology itself provides a number of classic tests, identifiers for classic tests, a univariate test, and also all the metrics associated with it, but also graph and plots that you can also tag to create semantic figures. So, of course, I'm presenting on behalf of the team, so this is all thanks to all these people that have been contributing and working very hard. That's really the teamwork that I've been presenting today. And really thank you for your attention, and I will take any question from here.
Thanks very much, Philippe. That was a really nice overview from going from the very basics of where on earth do I start with this data management business through to all, as you say, like the microservices that really help integrate different tools that people need to use together. So thanks for that. If anyone's got any questions, could you post the questions either in the question box or raise your hand and we can come to you? There seems to be a question from Alejandra. I'll just see if I can unmute you. Hello? Can you hear me, Alejandra?
Hello, can you hear
Hello.
me?
Yeah, we can hear you.
Oh, sorry, I might have pressed the button by mistake.
Okay.
So I'm in the same team as Philippe, so thank you.
Okay. That's fine. Okay, so I
No,
do. Pardon?
no, sorry.
You've got spies on making sure you're doing it right, Philippe.
Yes.
um, so a question from my side would be, so with respect to biosharing, you did mention that one of the things biosharing looks at is different types of minimum information models and how they're actually uptaken by uh, different repositories that actually store the type of data the minimum information models are meant to be applied to. One of the things I did wonder is what comes first? Do the databases start mandating the standards? Is that what makes the usage? Or is it that people start using it and then the databases pick up on this and then start to mandate it?
I think historically it's the first case. I think if you look back at Miami or MIAPI for proteomic or for microarray and proteomic respectively, the first the big groups assembled together and start working on it and identifying the key elements, and then each of them started implementing those requirements with their own formats, but the drive was the same. And then, of course, there is probably the other way around, that people start developing a resource, and then maybe some people start using the tools and they realize, okay, maybe you missed a few things, and then as a critic or as a request for improvement, they would come up with, okay, we need to do better.
Okay.
What we have not shown, and we are working on it, so I think it will be released maybe by the end of the year, is new visualization components to surface the content of biosharing in a more visual way, so people could see a bit more on graphical grounds what kind of information is actually and what kind of relation we have captured through the curation work of biosharing records.
Mm-hmm.
So yeah, there has been a huge amount of work that's been done this year by Peter McCulton and Alison on Alison Lister on improving the annotation and streamlining this, and the next bit of the work will be, of course, to exploit all those efforts.
Okay. And have you found a corresponding uptake in using of these in the community now that these extra tools are available for visualization? So I suppose you were generating carrots and seeing if people would come, I suppose.
Yeah, that's too early, I would say, to say at the moment.
Okay.
I think it will require a bit more time. There are still, I mean, the first 16 conference just took place, and there are still huge efforts around, identifiers and things like that. So to help cross-referencing resources to gauge acceptance. In our case, I think we still need to a bit more work to be able to really have kind of metrics of uptake. We can definitely see a wave of this kind of peak of following Miami of MI checklists that have been produced, and then it has receded a little bit. But I guess that we are about to witness also probably a round of reviews and that would highlight bits. Okay, it's time to revisit some of these checklists. Are they too stringent? Are they too sloppy? And so.
Yeah. I mean, as you say, this kind of area. I mean, all this area is quite in its infancy to some degree. It's we're working a lot on trying to get the community to see the advantages of it and how it supports them as well.
Yeah,
Um,
absolutely.
so with biosharing as well, you mentioned that there's different groups of people, obviously, that you target to help, and one of them was funders and journals. And this is something we don't always hear information about how researchers are trying to support funders and journals in a way. They're almost seen as the people who pay or the people who publish the research. So, what type of work or interest have you found happens with funders and journals with biosharing?
So it's a bit so they often come at least the roles come. So could you advise us which are the databases which are getting the most hits and which are the most popular? So they really sometimes come to us and say we are aware that we need to come up with a data sharing policy, and we would like to have a device to identify the key resources, those that are really highly regarded by the community. So that bit of information is definitely sought after.
Mm-hmm. Yeah.
And then from the funding agencies, well, I think that they, in a way, to assist them, and it's a similar use case. So they want to hear from us somehow which are the standards which they should know about, and so that they can start building guidelines or evaluation for the proposal that they are receiving to gauge the good value of data management plans produced forward by research scientists to have a feel for whether they've missed a key standard or not. And the other aspect that is interesting as well is that they also may want to know which are the gaps where they maybe be willing to play a role for supporting standardization efforts, because this is another bit of discussion that is interesting, is that standardization work is done, as I said, by two different bodies. Either it's grassroots scientists or it's a standardization body which is already established. But for grassroots scientists, it's not always clear whether this is a timely investment of their time and useful. So that's something that could be very useful to instruct efforts and reward scientists that are willing to commit resources to that.
Yeah, that sounds quite interesting. I've got a couple of questions from Stuart Owen, but he doesn't have any audio at the moment, so I'll ask them on his behalf. Okay. So the first question is, do you track any user metrics on biosharing, for example, common searches to identify where standards may be lacking or confusing?
So we do obviously record the logs, but we have not carried out deeply that analysis yet. So that's definitely something we would like to, we will definitely look into that, precisely to identify the gaps. Yeah.
Okay. Very good. That's good. And the second question is, how would you recommend using ISA tab if a study also comprises modelling steps or bioinformatics, as well as a typical lab assay?
Again, that's a very good question. I think this is where we organized a workshop last year to evolve ISA to incorporate the fact that to reflect two things. First, that data reuse starts to come online. People do not necessarily carry out new experiments; they would just assemble data sets from public resources and do some analysis. And the other bit is that people start doing simulation as well. So how do we record that in ISA? And of course, the other question is that the tabular layout has constraints. This is also why we evolved the serialization to have a JSON rendering. So it's almost, I would require almost a seminar, not a seminar to discuss to answer fully that question. But yes, I think also we can learn to delegate if it is about a data processing method, to delegate to the right tool. ISA was devised to begin with to, tell us about the experimental setup in your wet lab for your wet lab experiment. This worked reasonably well. Now, if you are doing for a very complex analysis process with branching and a lot of complex path, then of course we would probably hit the limits of the ISA envelope.
Mm-hmm. Okay, that sounds quite good. It also sounds like there might be a potential follow-up for us to have a targeted seminar at some point. So that's quite interesting. We're running out of time. In fact, we're already a little bit over time. So I think for now we can finish the questions. Thanks again for presenting, Philippe. It's been very interesting, and the questions Your answers to the questions have been very insightful. And if anyone has any questions that they want to ask outside of the webinar, or if you're listening to this on a recording, you can also go to the forums on the Fairdom website, which are hosted at Biosharing, and you can ask the questions there, and we can make sure that these are passed on to Philippe for further answers later on. And when you sign out of the webinar today, there'll be a short survey that comes up, so please fill in the survey if you can. So thank you once again, Philippe. It was a pleasure to have you on our webinar. And
thank you for having me.
You're welcome. And thank you everyone for turning up today. It was good to have you here again. And next month we won't be having a webinar again because it's a public holiday in the UK, so we will see you again in June. Thanks. Bye.
Good night.